Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about anomalous secondary growth in plants. So anomalous means abnormal secondary growth. It is not actually abnormal, it is actually deviation from normal or a variation. In many plants, the pattern of secondary thickening shows deviation from the normal type, and the term anomalous is given for this deviation actually. The anomalous secondary growth is more common in tropical plants, and the anomalous secondary thickening is not an anomaly or any disease in plants, rather it is an adaptation to suit the habit and habitat for the plant. So in vascular bundle, in dicot vascular bundles, the vascular bundles are conjoined, collateral, open because they have cambium and xylem and phloem present in same radius and arranged in a ring systematically. Formation of secondary tissues takes place by fascicular cambium. What is fascicular cambium? Suppose it is the stellar part and these are the vascular bundles. Xylems are present inner side and phloem to the outer side, and these are cambium, fascicular cambium between phloem and xylem. Okay, fascicular cambiums are present. From this cambium secondary growth takes place, and these are called fascicular cambium, and in the cortex core cambium is present, which helps in the formation of periderm. In monocot, vascular bundles are conjoined, collateral but closed. That means cambium is absent and are scattered throughout the ground tissue. They are not arranged systematically in a ring. And secondary growth is totally absent because they lack cambium. And cambium is the part where the secondary growth takes place, from where the secondary growth takes place. Any deviation in the above pattern of development in primary and secondary structure is called anomalous or abnormal secondary growth. Okay. So, what are the reasons for anomalous secondary growth? Normal cambium behaves peculiarly or irregularly, resulting in abnormal arrangement of vascular tissue. Normal cambium is situated in abnormal position, and normal cambium does not develop, okay, and if present is replaced by additional or accessory cambial ring. So sometimes normal cambium is there, but it is placed irregularly in abnormal position. Sometimes normal cambium is there, but it behaves abnormally, and sometimes abnormal cambium is present or additional layer of cambium are present. Okay. Next, abnormal or anomalous secondary growth is often observed due to abnormal position of cambium, abnormal activity of cambium, or abnormal position, as well as abnormal behavior of the cambium. So, it can be of two types. One is adaptive type of secondary growth, anomalous secondary growth. Second one is non-adaptive type of anomalous secondary growth. So what is adaptive anomalous secondary growth? If anomalous secondary growth has relation with habit or physiological function, such anomalous secondary growth is called adaptive secondary growth. They show such type of anomaly to cope up with the environmental condition. Okay. Next, non-adaptive type. If anomalous secondary growth has no relation with the habit or any external environmental condition, such growth is called anomalous growth is called non-adaptive type. Okay. Next, anomalous secondary. These are some examples of anomalous secondary growth. At first, anomalous secondary growth in Bignonia stem. Secondary growth in Bignonia stem is not typical of that found in dicot stem. it shows some deviations. In this plant, cambium is normal in position but show abnormal activity. At first, remember this point. Here, cambium is normal but they show abnormal activity. In some plants like Bignonia and other members of Bignoniaceae, the cambium is normal in position and activity for the first time, but immediately it cuts off different proportion of xylem and phloem at 4 alternative points in the form of a cross. The cambium produces secondary xylem and secondary phloem in variable quantities. In some segment, much greater amount of xylem is formed than phloem, and in others, more phloem than xylem. Okay, so in this way, it forms rich and furrow. That means like this, rich and furrow. Okay, so as a result, characteristic structure is formed with ridged and furrowed xylem cylinder. Ridges are wider than grooves. The secondary phloem formed in the grooves. These are grooves. Okay, and parallel stripes of bast fibers are formed. The cambium break off into small strips. The wider one remain on the top of the ridge and narrower one remain on the base of the groove. Okay. So, some cambium present on the top of the ridge. Some cambium present on the top of the ridge. Some are present in the base of the groove. Okay, like this. The number of wedges formed is usually 4 in number. Okay, 4 in number, symmetrically arranged corresponding to the position to the larger primary vascular bundle. This kind of adaptive anomaly exists in Bignonia because of its habitat, that is of a woody climber. So it is a woody climber. So they show this type of habit pattern or anomalous secondary growth. So these are phloem patches at 4 corners. So why Bignonia shows this kind of activity? At first, Bignonia is a climber. The stem grows around a support. Its inner side get compressed and outer side get stretched. Okay. Climber, the inner side gets compressed and outer side gets stretched. Okay, if hard tissues like secondary xylem remains in the central part of the stem during compression and stretching, the stem would break or crack. To avoid breaking or cracking, it requires some soft tissue in the central region. So central hard tissue is traversed by 4 phloem patches at 4 diagonal spaces. These are phloem patches. Okay. So there is direct relation between anomalous secondary growth and climbing habit of the plant. Hence, this anomalous secondary growth in Bignonia stem is described as adaptive anomalous secondary growth. Okay. Next, anomalous secondary growth in Boerhaavia stem. So at first, transverse section of Boerhaavia is circular in outline and reveals the following tissues from outside to inside. At first, epidermis is present, which is completely normal. Then cortex, it is well differentiated and consists of few layer of collenchymatous hypodermis, which is also normal. Collenchyma is 3 to 4 cell deep, but generally not near stomata. It is only one layer. Okay. Chlorenchyma is present inner to collenchyma. Chloroplast containing collenchyma. Next, chlorenchymatous cells are thin-walled and full of chloroplast, enclose intercellular spaces. Endodermis is clearly developed and made up of many tubular thick-walled cells. Inner to cortex. Then pericycle, inner to endodermis, pericycle is present, which is parenchymatous in nature. That means consists of parenchyma cell tissue, but at some places it is represented by isolated patch of sclerenchyma. Okay, next, vascular system. Here, vascular bundles are present in 3 rings. Generally, in case of dicot, 1 ring of vascular bundle is present. Here, 3 rings of vascular bundles are present. The innermost with 2 large bundles, in the middle ring 6 to 14 vascular bundles, and in the outermost ring 15 to 20 vascular bundles. Vascular bundles of innermost and middle rings are medullary bundles. Vascular bundles are conjoined, collateral and ender, which is normal, but total vascular bundles, all the vascular bundles arranged in 3 rings. 2 vascular bundles which are innermost are arc and large, oval and lie opposite to each other. Okay. So this is the diagram of transverse section of stem. Here the vascular bundles arranged in 3 rings. These are 2 rings. These are second layer. These are the second layer of vascular bundles, and these are the third layers of vascular bundles. So in this way the vascular bundles arranged in 3 rings. 3 rings, but normally all the vascular bundles are arranged in a single ring. So it is the anomalous structure of Boerhavia stem. But it is non-adaptive type. And this type of arrangement or anomaly is not an adaptation to any environmental condition. Okay. So next, anomalous secondary growth in Achyranthes stem. So Achyranthes stem, transverse section of Achyranthes stem shows following layers. At first epidermis, which is completely normal. Next cortex, okay. The main problem lies in the vascular system. So in young stem, the vascular bundles are conjoined, collateral, open and ender, but old stem shows secondary growth. Okay, in old stem, vascular system consists of primary phloem, secondary phloem, cambium, secondary xylem, conjunctive tissues, included phloem and primary xylem. So these all are present in the secondary stem. Okay, so primary phloem is crossed and obliterated. Secondary phloem is present in the form of complete ring and consists of sheath tube, companion cell, phloem parenchyma. A cambial strip is present between secondary xylem and secondary phloem. Secondary xylem and conjunctive tissues are undistinguishable. Large xylem vessels are very clear in thick-walled conjunctive tissues. In conjunctive tissues or prosenchyma are present in groups of included phloem or interxylary phloem. So there are phloem patches present in secondary xylem. Primary xylem present near the pith. Okay, in this way they show primary growth, primary xylem, next secondary xylem, and there are some phloem which are present in the secondary xylem. Next pith, it is well developed medullary rays. 2 medullary bundles are also present in the pith. Okay. Next, secondary growth and medullary bundle in the pericycle region, extrastellar cambium strips develop which produce secondary bundles. And cambium here also produce conjunctive tissue. Secondary vascular bundles and conjunctive tissue present without any sharp limit. So phloem of secondary vascular bundles appear in the form of patches. This phloem is called included phloem, and medullary bundles are leaf traces. So near pith, in the pith 2 medullary bundles are present. It is an abnormal feature. Next, some phloem patches are present in the secondary xylem. These are called as included phloem, which is another abnormal feature of Achyranthes stem. Next, anomalous secondary growth in Nyctanthes stem. So the outline of transverse section appear quadrangular. Okay, but normally it is circular, but here in Nyctanthes stem shows quadrangular appearance. At first epidermis, next inotuit cortex is present, then cortical vascular bundles are present. 4 vascular bundles are present in the cortex, situated one in each protruded bulge. Each conical bundle faces its pointed xylem end towards the outer side, and conjoined, collateral, open and exarch. These bundles may show secondary growth at maturity. So the stem of Nyctanthes is like this. So at four corner they have vascular bundles where xylem facing towards the outside and phloem towards the inner side. Xylem towards the outer side and phloem towards the inner side. Okay. So these are cortical vascular bundles. Generally in cortex, vascular bundles are not present. Vascular bundles are present in the stellar part. So this is an abnormal structure, abnormal feature in Nyctanthes stem. Then endodermis is not well developed. Pericycle is in the form of sclerenchymatous patches. Vascular bundle consists of primary phloem, secondary phloem, cambium, secondary xylem and primary xylem. Primary phloem is crossed and irregularly present in patches below the pericycle. Secondary phloem is present in the form of a continuous ring and consists of sheep tube, companion cell and phloem parenchyma. Okay. Pith is thin-walled and parenchymatous. What is the abnormality? The presence of cortical vascular bundle which are inversely oriented. That means xylem present outwardly and the phloem inwardly. 4 in number and never directly connected to main axial ring of vascular cylinder. This is one abnormal feature. The second one is these are leaf traces bundles. Cortical bundles have been also reported in some other families like Achyranthes, Umbelliferae, then Papilionaceae, Malastomaceae and Rutaceae. Okay, next. So this is the structure. These are 4 cortical bundles. So this is mirabilis and in mirabilis stem the anomalous secondary thickening occur due to formation of accessory cambial ring. There is one normal cambium ring. Apart from the normal one, there are many additional cambium ring exterior to primary vascular bundle. So here it is the abnormal thing in mirabilis stem. Okay. Next, Dracaena. Dracaena is a monocot and it is a typical example of anomalous secondary thickening in monocot stem. Typically, secondary thickening is absent in monocot. Therefore, secondary thickening itself is an anomaly in Dracaena, as Dracaena is a monocot. Dracaena young stem cross section, Dracaena young stem of Dracaena. Young stem of Dracaena shows some following structure. Epidermis is single layered, hypodermis is sclerenchymatous, closed collateral vascular bundle scattered throughout the ground tissue, xylem is endark. Anomalous secondary thickening in Dracaena is due to extrastellar cambium ring in monocot stem at the cortex, and cambium also show abnormal activity here. Okay. During secondary thickening, the formation of secondary meristem or secondary cambium occur in inner region of parenchymatous cortex. Okay. The activity of cambium is abnormal. How? It produces secondary vascular bundles on its inner side only and parenchymatous cells towards the outer side. The presence of cambium is abnormal in monocot because generally cambium is absent in monocot. So presence of cambium is abnormal again. Here cambium also shows abnormal activity. It produces parenchymatous cells towards the outer side and vascular bundles towards the inner side. The secondary vascular bundles are amphivasal where phloem is surrounded by the xylem. Okay. Next, later cambium produces more parenchymatous cells towards the inner side that pushes newly formed bundles toward the center. The activity and position of vascular bundles keep on changing like this. Sometimes it produces parenchymatous cells towards the outside. Sometimes it produces parenchymatous cell towards the inside. The activity and position in this way keep on changing. The second ring of vascular bundle is alternating in position with the first ring. The vascular bundle in last inner ring are embedded in a mass of lignified conjunctive tissue. Cork cambium activity is normal and produce cork and secondary cortex at the outer region, that means in the extracellular region. So it is the structure of Dracaena stem. Okay, here it is the cambium. These are amphivasal vascular bundle. This is cork cambium. So in this way, different plant species show different anomalous secondary growths. So I hope you really enjoyed the session and definitely get benefit from this video. And if you liked it, please hit the like button. Please share with your other friends so that they also get the benefit from it. And please subscribe to my channel Sunanda's Tutorial. Thank you.